Well, welcome to this seminar in the series of the Department of Epidemiology of the Harvard Chan School. My name is Bert Hoffman. I'm the chair of epidemiology, and I welcome you here at 1 p.m., but for some of you it may be later, and for others it may be earlier. Great pleasure to introduce today's seminar speaker, Dr. Stefania Papatheodorou. Stefania is Associate Professor in Epidemiology at Rutgers School of Public Health. She is trained in obstetrician and gynecology, obstetrics and gynecology, I should say. She has been and still is appointed also in our Harvard School of Public Health. She is an adjunct associate professor of epidemiology in our department. And it's wonderful again, Stefania, to have you here. You have been quite a number of years more directly with us, but it's great to see you. And it's also great how you do well at Rutgers. Stefania is working on, and you will see that I'm sure in the seminar, on the intersection of extreme weather, air pollution, pregnancy, and children's health. And she uses a wide range of data sources, administrative or otherwise, electronic health records, birth registries, claims data. And currently your emphasis is on effects of prenatal and early childhood exposure to extreme weather on neurodevelopmental outcomes. And that is then the topic, or at least in a more general sense, the topic of your seminar, From Signal to Noise, Addressing Multiplicity Using the Exposome Neurodevelopment Example. My great pleasure to give the floor to Dr. Stefania Papatheodorou.
Thank you very much, Dr. Hoffman, for the very kind introduction, and thanks to everyone for being here. It's such a pleasure to be here, and thanks for the invitation. So I'll just start sharing my screen to begin with. So the title of this talk is From Signal to Noise, Addressing Multiplicity Using the Exposome Neurodevelopment Example. And I will be using the neurodevelopmental outcomes as a case study to highlight how we can more rigorously address multiplicity while still capturing the complexity of environmental exposures across critical windows of development. So we'll get started by discussing about multiplicity. So in epidemiology in general, we don't test one exposure and one outcome only. We are often evaluating multiple exposures, such as multiple medications, multiple pollutants, multiple environmental chemicals, several adverse outcomes, several adverse pregnancy outcomes, several adverse childhood outcomes, and different exposure windows. And this all happens simultaneously. And this naturally inflates the number of tests we are applying, raising the likelihood of false positives. And multiplicity isn't just a statistical nuance. It is one of the key reasons why findings in our field can be difficult to replicate or synthesize for policy. And then our challenge is how to balance this exploration, which is at the heart of scientific research, with statistical rigor, and that's what we are going to explore today. So when we think about environmental health, exposures are never isolated. They originate from multiple mixture sources like traffic, industry, wildfires, and they translate into external exposures that we can measure at the neighborhood or the regional level. And from there, these exposures enter the body and become internal exposures, influencing molecular targets and biological pathways that can lead to tissue effects. And ultimately, these changes manifest as individual health outcomes, which then aggregate into population-level outcomes that we study in epidemiology. And each of these stages can be measured, and each one introduces multiple analytic choices. And that's why the exposome research, which is the totality of exposures from when we are born until we die, is powerful, but also it's prone to the problem of multiplicity. It generates many potential exposures, pathways, and outcomes to test. So As we all know, data streams in public health are getting much larger and more complex. We are no longer working with just a few hundred or even a few thousand participants. These datasets now can span hundreds of thousands of individuals with millions of genotypes and thousands of phenotypes. For example, the UK Biobank provides data on over 500,000 participants, 1 million genotypes and thousands of phenotypes with genomic data linked to rich health, environmental, and lifestyle information. Projects such as the HEALS and Exposomics in Europe are generating high-resolution exposome data, allowing us to study environmental exposures across the life course. The HELIX project, for example, is focusing specifically on early life exposures and child health outcomes. The CHEER program here in the US has built critical infrastructure for children's health exposure analysis. At the same time, our capacity to measure exposures and compute complex models has grown tremendously, producing high throughput data streams that enable unprecedented insights, but also amplify the multiplicity problem. And together, all these initiatives demonstrate that our capacity to measure exposures and compute complex data is now truly high throughput. And this creates both new opportunities for discovery, but also brings unique challenges in terms of study design, interpretation, and causal inference. So as exciting as exposome research is, there are still major challenges that we need to address to make it robust for discovery and causal inference. First, we have to think about the scale. We can generate thousands of hypotheses when we are measuring so many exposures simultaneously, along with so many outcomes. This leads to a multiplicity of hypotheses problem. Essentially, the more things we test, the more likely we are to find false positives. Second, big data in this field are almost always observational, and that means we face a multiplicity of biases problem. These include classical issues like confounding, selection bias, or even reverse causality, which can make it very difficult to establish whether exposures are truly causing the outcomes we see. And finally, we have to ask ourselves, is the high throughput exposome really ready for prime time? Even if we can measure these hundreds of exposures simultaneously, we need to be certain that we can identify what our signals really mean. And one of the major reasons findings fail to replicate in environmental epidemiology and many other fields, not only environmental epidemiology, is multiplicity. And what this figure shows is that when studies have small sample sizes or imprecise measurements, like in the panels on the left, results can vary widely just by chance alone. And out of the many tested strategies or exposure metrics, we tend to highlight the one that looks significant. That's the effect of selective reporting that is shown in orange. But when someone tries to replicate that same result, the next study estimates can bounce all over the place because the signal was not real. It was just noise that was amplified by this multiple testing and small numbers. And on the right side, with large samples and more precise measurements, that scatter tightens. The estimates converge around the true value and replication becomes possible. So a key message is that multiplicity, whether it's from multiple exposure windows, multiple outcomes, multiple models, or subgroups, it can easily manufacture apparently significant findings unless we explicitly control for it or pre-specify hypotheses when possible. And in recent years, the scientific community has been rocked by the recognition that research findings often do not replicate on independent data, leading to what has been referred to as a replication crisis, reproducibility crisis, or statistical crisis in science. And in particular, a series of attempts to reproduce results of published research findings in different disciplines found that these replication efforts produced much weaker evidence than the original study. And this crisis has consequences far beyond an insular world of scientists. Experts with strongly disagreeing viewpoints and publicized results that are subsequently contradicted are highly detrimental to the trust of the general public and decision makers have in scientific results. So the air pollution, ambient exposome, neurodevelopment context is a very good example to illustrate multiplicity. Because we're not looking at one pollutant and one disease. We are looking at a mixture of correlated exposures, such as particulate matter of less than 2.5, fine particulate matter, nitrogen dioxide, ozone, other pollutants that are acting through overlapping biological pathways. At the same time, the outcomes, such as autism spectrum disorder, attention deficit hyperactivity disorder, language and learning delays, and others, are also interrelated. Sometimes they co-occur, often sharing some developmental roots. Add to that the question of when exposure matters most, which is a very important question in environmental epidemiology. Is it preconception? Is it during specific weeks or specific trimesters? Is it early postnatal life? We end up with hundreds or even thousands of possible combinations. And this makes it a good case study to think about multiplicity, not as a statistical nuisance, but as a reflection of biological and environmental complexity that needs thoughtful design and interpretation. And we all know that diseases like autism spectrum disorder and ADHD have been on the rise. That's why there is so much awareness in the scientific community. And here on the left, you can see the dramatic rise in autism prevalence from about 1 over 150 children in the early 2000s in about 1 over 36 children in 2020. And on the right, we see the data for ADHD. It shows a similar trend with both diagnosis and medication use increasing steadily since the late 1990s. And these patterns reflect not only improved awareness and diagnosis, but also raise questions about environmental and social factors that may contribute to the risk. And because these disorders are multifactorial, involving genetic, social context, multiple environmental exposures over sensitive developmental windows, they provide an ideal test case for examining how we handle this multiplicity, multiple exposures, multiple correlated outcomes, and multiple time windows of vulnerability. And someone can say in outcomes like birth defects, we can pinpoint the week based on biology on when we would expect a detrimental effect to happen and compare it with an effect, a time period where an effect is not expected to happen. Birth defects happen very early in pregnancy, so we would not expect an exposure that happens in the third or the second trimester or any late weeks of pregnancy to have an effect. But this is not the case in other research questions, like the brain development. So here in this slide, we see how brain development unfolds across gestation and early life. So in the first trimester, the brain is laying its foundation with proliferation and neurogenesis, meaning new brain cells are being created, early cell migration is beginning. By the second trimester, those processes continue, but we also see neurite outgrowth, the start of the brain's wiring. During the third trimester, synaptogenesis takes off. This is when connections begin between neurons form and synapse function begins to mature. And once we reach birth and early childhood, the focus shifts towards building and refining the neural networks, which continue to develop well into the first years of life. So the timeline underscores why both prenatal and early postnatal periods may be so sensitive to environmental exposures. And any disruption during these windows can have lasting consequences for brain function and neurodevelopment. Also, whenever we are approaching a research question, it's always important to have in mind what prior studies have shown. So when we're trying to answer the question about the association between air pollution and autism spectrum disorder, we would need to see what prior studies have shown on this topic. So here we see the results of a recent systematic review and a meta-analysis. And on the left, you can see a forest plot that summarizes studies on different pollutants, including PM2.5, PM10, nitric dioxide, and ozone. And what we see here is that many studies shows odds ratios above 1, particularly for fine particulate matter and nitrogen dioxide, suggesting some elevated risk. But we also see substantial heterogeneity across the studies, which reflects differences in exposure assessment, study design, and populations. On the right, the forest plot is stratified by timing of exposure, prenatal versus early life. And this allows us to ask whether the critical windows of exposure differ. So we again have some studies suggesting that prenatal exposure has stronger associations, while others find postnatal exposures equally important. And again, variability is high and the confidence intervals overlap. So this evidence supports the plausibility of a link between both prenatal and early life air pollution exposure and autism spectrum disorders, but also underscores these methodological challenges: heterogeneity, multiplicity of comparisons, and timing of exposure. Here are the results from recent meta-analysis again for air pollutants and ADHD. And on the left, we see the prenatal exposures. We see several studies that report associations between PM 2.5, nitrogen oxides, and ADHD risk. The effect sizes are small to modest, but we do see a consistent picture of increased risks. On the right, we look at the postnatal exposures, and this picture is somewhat more variable with wider confidence intervals, but we do have many studies suggesting that children exposed to higher levels of these pollutants in early life may also have higher ADHD risk. And this comparison is telling us that there may be some shared pathways between these diseases and other neurodevelopmental delays. For example, inflammation, oxidative stress, disruption of neurotransmitter systems that underlie both disorders. And at the same time, the differences we see hint at disorder-specific mechanisms. Autism may be more tightly linked to disruptions in the prenatal period, while ADHD may reflect both prenatal programming and cumulative exposures during childhood. So I will show the results from studies that we have been performing using a mother-child pair cohort that from Medicaid data that was created from colleagues at Brigham and Women's Hospital and Harvard. And this is a slide that shows how this cohort has been created. I'm not going to go into a lot of details, but what you can see here is that we have to apply certain eligibility criteria from the identified pregnancies. And we want to have a good coverage before last menstrual period and sometime after delivery to have a population that would be representative of the Medicaid population. We used exposure data. So our data set spans from 2001 to 2014, around 1.8 million mother-child pairs in these populations with small differences between the pollutants, but overall the number is approximately the same. With atmospheric exposures, we used well-validated models for all the pollutants, and we aggregated them at the zip code level because this was the spatial resolution of the health data that we had, and we averaged the ones that we had the ability by week. And we also took into account residential mobility during pregnancy. So we had we assigned different exposures, weighted exposures for people that moved during pregnancy, having a different zip code at last menstrual period and a different zip code at birth. The outcomes have been validated from the Medicaid claims data using algorithms that have very good positive predictive value, generally very high, ranging from 82% to 98%, suggesting that these claims-based algorithms can be used to study neurodevelopmental delays quite accurately. Again, I'm not going to go into the details of what we adjusted for and what data we had, but we had rich information about personal level covariates, maternal age rates, race, education, all the things that were included in the claims data. We had several variables at the zip code level that can characterize area level socioeconomic status, for example, very well, some at the county level. And also for our analysis, for most of our analysis, we used Cox hazards models, starting from birth and ending at when the outcome happened at death or disenrollment from Medicaid. We use the distributed lag models, which are very popular in pregnancy research by gestational week. And we also applied, along with the distributed lag models, inverse probability weighting for to see the possibility of bias due to loss to follow-up. And we also applied generalized propensity scores to address residual confounding. So these are the results regarding residential greenness, which is a protective factor, is assumed to be a protective factor for neurodevelopmental delays. This is the work of Dr. Hyeon Choi, which was published earlier this year. And here we, on the right, you see what happens when we have mutually adjusted all exposures and outcomes. We have 3 different windows of exposure: preconception, pregnancy, and postnatal. And on the left, you see what happens without mutual adjustment, on the right, with mutual adjustment. So in general, we see that exposure to residential greenness is protective for most neurodevelopmental delays, which is something that is intuitive and makes sense. There is some specificity in some of the results. For example, we do see that for autism spectrum disorder, this protective effect is highlighted only during the pregnancy period. So there is some specificity of these protective effects. And the big takeaway here is that the access to green space may be a modifiable protective factor for children's neurodevelopment, though the strength of the association depends on how we handle these correlated exposures and outcomes. Here I'm showing the results of a distributed lag model paired with Cox hazards model. This is the work of Dr. Matthew Sapler. And here you see on your left, you see the preconception and the prenatal results. And on the left side, we see the postnatal results. These are distributed lag models, and the graphs are showing on the x-axis the gestational age from preconception, not only the gestational age, but on the slides that include the preconception period, you also see the preconception period. And on the y-axis, we see the hazard ratios. So what basically stands out from these graphs is that, for example, for ASD, the results suggest some possible vulnerability during pregnancy with a hazard ratio between 23 and 31 weeks, showing a 9% increase in hazard per 10 micrograms per cubic meter, while the hazard ratio for the overall pregnancy period was 1.17 with 95% confidence intervals ranging from 1 to 1.35, 37, sorry. Here are the results for gaseous pollutants and neurodevelopmental outcomes. This is the work of Dr. Wanyu Huang, and this is only preconception prenatal periods. On the right, we have nitric dioxide. On the left, we have ozone. And here we see that generally these effects, no matter the preconception or the prenatal period, we see that they are hovering at the null value. We do not really see some sensitive windows of exposure happening. Here are the results from the previous analysis that I just showed for the postnatal exposures. And again, for nitric dioxide, the results are mostly null, apart from learning difficulties that show an 18% increase in the hazard. And ozone, the results are hovering around 1. Here we're looking at the effects of prenatal exposure to wildfire-related PM2.5 on autism spectrum disorder. And here you see the lag-specific hazard ratios for autism spectrum disorder across the gestational weeks. And this curve suggests an elevated risk around weeks 19 to 25 with a hazard ratio of 1.17, meaning about 17% increase in risk per 1 microgram per cubic meter increase in wildfire-specific PM2.5 during this window. When we look at the cumulative exposure across the entire prenatal period from week 1 to week 33, the hazard ratio is around 1.11. And here we see the results stratified by sex. So among males, you can see a similar pattern with increased hazard ratio during the mid-gestation, while among females, the curve remains relatively flat with no clear signal, but we have to take into account the difference in the sample sizes because autism spectrum disorder is much more prevalent in males than in females. And this suggests that mid-pregnancy may be a particularly sensitive period in terms of neurodevelopmental vulnerability to wildfire smoke exposure. And these findings do highlight not only the timing of exposure, but there may be also potential sex-specific differences in susceptibility, which have been also noted in other environmental exposures. These are the results for ADHD. And the graph here shows the overall lag-specific effects. We see that exposure between weeks 21 and 30 of gestation seem to be the most critical, with a hazard ratio of 1.1 per 1 microgram per cubic meter increase in wildfire-specific PM2.5, and the confidence intervals range from 1.03 to 1.20. And this suggests a modest but probably an important increased risk during this window. And when we look at the cumulative exposure across the entire 1 to 33-week prenatal period, this effect is weaker, is 1.07. Breaking the results down by sex, here we see for males, there is a gradual increase in risk across gestation, whereas for females, the curve is flatter, but this probably is an effect of the smaller sample we have for females. And these findings suggest that mid to late prenatal exposure to wildfire smoke may represent a critical window for ADHD risk and ASD risk. So some of the toxicity of the wildfire PM2.5 specific exposures may be acting the same way to manifest both diseases. So here the challenge we face when trying to estimate these dynamic effects using these distributed lag models is that we are looking at 7 different exposures at least. We may look at more than them, but we already have at least 7 exposures. Exposures like fine particulate matter, nitrogen dioxide, ozone, temperature, humidity, other weather variables, wildfire smoke, protective factors such as residential greenness, or other factors like neighborhood deprivation. On the outcome side, we're examining 7 neurodevelopmental outcomes: autism spectrum disorder, ADHD, learning difficulties, speech and language disorders, developmental coordination disorder, intellectual disability, and behavioral disorders. And these exposures don't just matter at one point in time. We focus on 3 critical developmental windows, some of them having a weekly resolution, some of them having an annual resolution like a postnatal period. And we end up, if you put all this together, 7 exposures, 7 outcomes, and 49 possible windows of vulnerability, this results in more than 2,300 comparisons. So the scale of the problem is large, and it illustrates why teasing apart meaningful signals from all this noise is so methodologically challenging. So how do we deal with this problem? Several strategies have been proposed. First, we have the classical approaches like Bonferroni or false discovery rate adjustments. And these are simple and widely used, but the downside is that they can be overly conservative. That means while they control for false positives, they may also cause us to miss real effects. The Bonferroni correction remains the most widely used method for controlling for family-wise error rate. However, this is often criticized for being overly conservative, increasing the risk of false negatives, and potentially overlooking meaningful effects. There are other similar methods, like the stepwise methods, such as the Holmes procedure, adjusting significance thresholds sequentially, or the Hodgberg method, ranking hypotheses to set critical values. They do offer a more balanced approach by controlling type 1 error rates while maintaining the statistical power. And these methods are generally preferred for their ability to balance stringency and flexibility. Another approach is penalization and shrinkage methods using machine learning algorithms like lasso, elastic net, Bayesian shrinkage. These techniques help reduce the noise in high-dimensional data and can help stabilize estimates when we are looking at many exposures at once. Also, there are mixture modeling approaches, such as weighted quantile sum regression, quantile G computation, Bayesian kernel machine regression. And these are very powerful because they explicitly capture correlations and interactions across multiple exposures, which is very relevant in an exposome framework. But they don't always answer the question we are interested in answering. For example, sometimes we're not interested in the mixture as a whole, but we are interested in whether a specific pollutant is harmful so that we can address the sources of that pollutant. And finally, we can leverage the lag structures using models like distributed lag, nonlinear models, Bayesian distributed lag interaction models, coupled with causal inference methods such as G computation, inverse probability weighting, or propensity scores. That would allow us to examine how timing and duration of exposures matter, which is crucial when we think about sensitive developmental windows. There isn't just one perfect method. Each method comes with trade-offs, but together they give us a toolkit for addressing multiplicity in this complex environmental health data. Another point I want to make in specifically questions that have to do with multiple chemicals, multiple pollutants, is whether it is appropriate to perform multiple testing corrections. So when we evaluate several air pollutants and multiple neurodevelopmental outcomes, it may look on the surface as if we are multiplying the tests and we are inflating type 1 error. But in fact, these are very distinct scientific hypotheses. Pollutants like PM2.5, NO2, ozone, wildfire smoke are actually different exposures with different sources, different exposure patterns, and different toxicological evidence. So each carries its own prior hypothesis about how it may contribute to neurodevelopmental risk or other health risks. Similarly, outcomes such as autism spectrum disorder, ADHD, they may represent biologically distinct processes. We are not asking the same question multiple times. We are interrogating different potential mechanisms. And in this context, applying just broad multiple testing corrections would be overly conservative and we would risk obscuring some true associations. And instead, we need to emphasize the hypothesis-driven analysis, biological plausibility, and consistency across pollutants and outcomes. And that approach avoids treating careful etiologic research as though it is exploratory data dredging. A few things about the distributed lag models that have been so popular, and I just showed you some results, but if you look at any time-varying exposure during pregnancy, I think the majority of the papers are using these models. So these models actually didn't start in epidemiology. They started from econometrics back in 1960s. The first formal version was the Almond lag model, introduced by Shirley Almond in 1965, which was designed to describe how the effects of an exposure, in her case, the capital investments, were distributed over time. The idea later was made its way into environmental epidemiology in the 1990s from researchers at the School of Public Health, at the Harvard T.H. Chan School of Public Health, when researchers began using distributed lag models to study short-term air pollution effects on mortality. And a breakthrough that happened around 2010 is when the nonlinear models were developed and relevant software was distributed and relatively easy to use. And this extended the concept to handle both nonlinear exposure-response relationships and complex lag structures using splines and cross-basis functions. And since then, these distributed lag models have become a core tool in environmental health research, especially for studying time-varying exposures like temperature, air pollutants, during pregnancy, where we have several vulnerable windows of exposure when both timing and dose matter. So here is one of the big challenges we face when we are using the distributed lag nonlinear models and that have become so popular in the literature. These models are descriptive tools. They are incredibly flexible in showing us how the exposures vary across different lags, but by themselves, they cannot actually identify causal effects. And this is a limitation that we need to keep in mind. Another issue is the multiplicity of estimates. Every lag and every exposure value generates an estimate. And put together, you may end up with this huge high-dimensional surface that's full of correlated comparisons. And this may lead to the final point, which is the risk of overinterpretation. So without careful correction, it's very easy for investigators to pick out just a few lags of exposure windows that look significant and then highlight only those without even discussing about the global effect or the possibility that this may be due to chance. That can inflate the type 1 error rate and can give us misleading conclusions if we're not cautious. So overall, while the distributed lag models are a powerful descriptive tool, they do require careful handling to avoid drawing causal claims that they cannot actually support. But they can become very powerful when they are coupled with causal inference methods. So here is an example. This is the work of Dr. Michael Jung. We have extended this distributed lag model using G computation to simulate counterfactual exposure scenarios, essentially asking, what if weekly exposure levels have been lower throughout gestation? And here we applied this example using preterm birth as an outcome. And here we see the workflow that summarizes the steps. So we need to format the data into a long person week structure so that each observation represents a specific gestational week. We then fit a distributed lag model under the observed exposure pattern to estimate the weekly risk of the outcome. Here, as I said, is preterm birth. And the third step simulates a hypothetical intervention, for example, a 5 microgram per cubic meter reduction in PM2.5 each week, and then recomputes the weekly conditional probabilities under that counterfactual exposure. And the final step is to aggregate these weekly specific risks to produce an overall cumulative risk curve that compares it to the observed scenario. So here we see the results. The solid line is the observed risk of birth by gestational week, while the dashed line represents the simulated lower exposure scenario. And the difference between the two corresponds to the estimated causal risk reduction under that intervention. So an approach like this would combine the temporal flexibility of the distributed lag model with the causal interpretability of the G computation, allowing us to estimate what would happen if we would shift the exposure distribution during pregnancy. I want to spend like a couple of minutes discussing another concept that has gained a lot of popularity in other fields like pharmacoepidemiology and clinical epidemiology, which is the target trial emulation. It is very influential in certain fields, but it has very serious limitations for environmental exposures. And here's why. So in the target trial emulation, you would need a clear point when individuals are assigned to a treatment or control, even if that assignment is emulated from observational data. But for environmental exposures, no one is randomized or assigned. Exposures vary with geography, season, weather, and pollution sources. And this makes it difficult to define a meaningful baseline time at which exposure is initiated versus withheld. Then drugs or interventions are usually binary. You have the treated population versus the untreated population, or categorical if you're looking at those levels. Environmental exposures are continuous, time varying and unavoidable. Everyone is exposed to some level of air pollution or temperature or other exposures. And that means that there is no true untreated group to emulate, only relatively lower versus higher exposures. Timing is ambiguous. Target trials require clearly defined time zero, the date of randomization, start of the treatment, but it's very unclear what is time zero for environmental exposures. Would it be the first day of pregnancy? Would it be conception? Would it be each day or hour of exposure? And because exposure accumulates continuously, defining eligibility, treatment assignment, and follow-up in a trial-like way is often artificial. Also, in a drug trial, one participant's treatment does not affect others' outcome. But for environmental exposures, like air pollution and across neighborhoods, for example, they do violate this assumption because you cannot really assign one group to low PM2.5 without affecting the nearby populations. And of course, the exposure is not controlled by investigators. The target trial emulation assumes you can, in principle, randomize the intervention, but we cannot randomize or manipulate wildfire smoke, urban heat, regional ozone levels. And this is important because we do see several applications of the target trial emulation without really thinking through whether the assumptions and the actual methodology is applied. There are better suited alternatives for environmental epidemiology, like quasi-experimental designs, natural experiments, difference in differences, interrupted time series. The case crossover design for acute exposure is a powerful design for transient exposures. Distributed lag models to capture the cumulative effects paired with causal inference methods like the ones that we just discussed about, G methods, marginal structural models for time-varying exposures. So addressing multiplicity is not just a statistical issue. It's also about how we communicate our analysis and interpret the findings. And first, the pre-specification is key. We should clearly define our hypotheses, exposures, and critical windows if possible. Sometimes it's not possible, as I showed for neurodevelopment, before running the analysis. And that would help distinguish what was planned from what was discovered post hoc. Second, we need to separate different exploratory from confirmatory work. Exploratory analyses are very valuable for hypothesis generation, but they should not be presented as causal evidence. Also, graphical reporting can enhance transparency. Tools like exposome-wide association plots, or DAX, make the analytic structure very visible. And that can help reviewers and readers see where multiple testing happens and where correlations may occur. And also, language matters, using phrases like associated with versus caused by signals appropriate caution, especially in these high-dimensional observational studies. And ultimately, good communication is really part of good science. It helps readers understand what is truly robust and what is still very uncertain. So looking ahead, there are several ways we can strengthen the reproducibility and credibility of environmental epidemiology. We can triangulate with triangulation of evidence, combining different study designs and analytic approaches, and replicating findings across populations is going to be very important. We do know that not single data set or model will ever be definitive, but convergence across independent sources builds causal credibility. Simulation studies can help quantify the extent of multiplicity and the risk of false discovery under realistic data structures, and they can give us a way to stress test our analytic choices before we apply them to real data. Third, very important, open science practices, transparent analytic pipelines, pre-registration, and code sharing are very critical for making our work reproducible and testable by others. And finally, policy relevance. We need to move beyond very narrow statistical significance and focus on what is really meaningful for public health, effect sizes, population impact, and potential for intervention. And progress will not depend on just better models, but on a culture of transparency, replication, and scientific humility, I would say. So to wrap up, multiplicity in some scenarios is unavoidable, but it's also manageable. The scale of exposome research does force us to confront this directly rather than pretending it's not there. If we use transparent methods, pre-specify key hypotheses, and communicate results with appropriate caution, we can transform what might look like an analytic noise into some credible scientific signal. And the broader point is that methodological rigor isn't just an academic exercise. It is what underpins public and policy trust in epidemiology. And the challenge isn't to simplify the complexity, but it is to handle it honestly and systematically. So with that, I want to thank you for your attentions, and I would be happy to take any questions.
Dr. Papatheodorou, thank you very much, Stefania, for a great overview and mentioning many thorny, may I say that, issues. Yes, there you have applause here, I see.
Thank you. Thank you very much.
And rightly so. May I invite questions or comments from the audience? Whom do I give the floor? Please open your screen or speak up directly or have a question in the chat. That's also possible. Let me have a little look. There's a question, I cannot really see the name, but if, I start with this one, if neurodevelopmental delays are heterogeneous and likely follow non-proportional hazards, have you considered accelerated failure time regression?
Yeah, this is a fantastic question. So thank you very much for this. I was not able to go into the full details of our analysis plans, but we did. One important thing that I didn't discuss is the computational burdens that all these methods have. So to answer your question, yes, we have a considered accelerated failure time regression, which does not assume proportionality of hazards. We finally, and we also applied stratified Cox models, which we allowed the baseline hazard to vary by year, by sex, by some of the variables that were violating the proportionality of hazards. They gave similar results, so we presented the stratified Cox analysis in our results. These accelerated failure time regression models may take too long to run, so sometimes when you do not have the computational capacity, it may be very, very difficult to apply them. We did apply them with, anyway, the results were comparable. We presented the stratified the stratified Cox models. Now, the second question, in the mutual adjustment models, how was the risk of over-adjustment and collider bias through conditioning on what may be mediators? So we discussed about this question a lot. We did apply G computation methods to make sure that our results were not, we did not capture the same variance by using multiple windows of exposure. And results were similar with these mutual adjustment models. The point I want to make with these questions, which are fantastic questions, is that there is some challenge on applying all these complicated methods together, along with computational burdens of having almost 2 million observations. So sometimes things take too long to run. I'm not saying we will compromise what we're doing because of these computational problems, but we need to take them into account. And the other is how do you describe all these methods in a paper without putting all this information in a supplement? So another challenge when we are trying to present the results is that we can end up with a fragmented literature. One paper is going to be for PM2.5, another paper is going to be, which happens inevitably, but the fragments and how you know, where we're going to end up with the separate pieces of information we convey needs some thinking. What we are doing is that when we apply a method that is addressing causality, like the inverse probability weighting or G computation or the generalized propensity score, we put this information in the supplement because there's not enough space in the papers.
Thank you. The questions, these great questions, were from Marie-Céline Schulte. Marie-Céline, would you like to follow up? Or do I put you now on the spot? Please go ahead, Marie-Céline. You opened, so that's nice.
Thank you so much for your incredibly thoughtful presentation of your work and equally thoughtful responses to my questions. I am working with accelerated failure time models and sequential causal mediation models. And it's true, your point about the careful thinking about what stays in the main paper versus what goes into supplementary materials. I'm still working through that on a paper right now. So yeah. I mean, I have managed to consolidate findings, especially on sequential causal mediation models, because you can see the cumulative proportions mediated, et cetera, and the, you know, all the different direct and indirect effects, depending on what kind of outcome measure you're looking at. But I managed to do that. And the accelerated failure time models, it's true that they can take a bit of time. I'm working in the UK Biobank, and yeah, all of it takes time. That's true. And computational capacity and funding. Fortunately, the biobank does have some support for computational costs, depending on your profile as a researcher. Yeah, where you come from. And yeah, so thank you again.
No, these are really fantastic questions, because these are issues that, this is, at least I'm presenting what we are doing. I'm not saying this is the best thing to do, but this is how we address those challenges. I'm open to hear more recommendations from others that, found a better way to do this.
Thank you. Any further questions? Can I ask one? Take the liberty to ask a question. In fact, we do not have so much time anymore, but you say multiplicity is a major issue, but you also said more or less in passing, well, we can perhaps restrict our questions to, say, one element of the exposome and one outcome, and thereby perhaps reduce a little bit of the overwhelming multiplicity issues. So why is that not a reasonable approach? Is that too naive, what I say?
It's not naive at all, and it is very plausible in questions that have a very, very clear biological pathway. We know the timing, we know the biological processes. For certain birth defects, for example, we even know the days that we are expecting these to happen, right, in the course of a pregnancy. So we can apply biologically driven hypotheses in all these questions that we are asking, but this is not always possible. And this is actually one of the strengths that, for example, the distributed lag models present, that it is a data-driven approach that will eventually show what is the critical window of exposure. This is not possible in outcomes like neurodevelopment, where any of these windows may be equally sensitive and vulnerable, and we want to provide the correct information. So inevitably, we have to test all of them and see what they are providing. I think that it is very important to present all the results in a scientific paper like this or any work, and show all the results for the entirety of the test that you applied, the windows that seem to be sensitive. Otherwise, the problem arises when selective reporting comes into play and highlighting only the important parts without telling the full story.
Yes, yes, I understand. Yes, thank you. Thank you very much for that answer. Well, I think we are in time. We have to change. Students have to have the opportunity to go to another class, so we have to stop here. But I would like, first of all, to thank you, Stefania, very, very much for an enlightening seminar. I thank all of you who were here in this afternoon, at least on Eastern time scales. And I already announce next week's seminar, same time, same place, again on Zoom. It will be at the interface of epidemiology and health decision science. It's about cancer screening, various approaches in cancer screening, by Dr. Uwe Siebert. And we look very much forward to that. But that said, I thank again very, very much Dr. Stefania Papatheodorou. Many, many thanks, and see you.
Thank you. Thank you very much for the invitation and for all of you that you were here. Thank you.